What is a meta-analysis? I want to start with a citation from Glass, who more or less invented meta-analysis, and he summarized meta-analysis as follows. Meta-analysis is a statistical analysis of a large collection of analysis results from individual studies for the purpose of integrating the findings. So let's look deeper into meta-analysis. Here is basically a broad overview on which data one might analyze during a research endeavor. First, we can talk about primary analysis. So basically that's an analysis of data you all should know, because it simply means analyzing data for the first time. So let's assume you're starting a research project. For example, you're conducting a survey, you're asking people some questions, and afterwards you analyze your data and gather your results. That's basically exactly what a primary analysis is about. So what is a secondary analysis? A secondary analysis means analyzing data that has been analyzed before, for example, to test new hypotheses. So let's assume you, as I said, you conducted your survey, you already performed an analysis, and then you decide, maybe there's something more in the data which I missed, for example. So you go back to your data and you analyze it again, but you test, for example, for different hypotheses or for a different model, for a different structured equation model, for example. So you do a secondary analysis because it's a different kind of analysis. It was not the primary purpose why you collected the data. So your goal or your aim is different than your reason why you collected the data in the first place. So this were primary and secondary analysis, and meta-analysis is a bit different because it means an analysis of an analysis. So it basically is described as integrating findings from several studies by combining the results of separate studies. So what you do is that you don't conduct a survey on your own, but you conduct primary studies. So you, for example, search in a literature database for studies that have already conducted, for example, a survey on a particular topic you define. And you look at those studies. These studies are called primary studies. They all did a similar kind of analysis, or at least provide a similar set of data. And you integrate those findings from your primary studies and build up your own database to run your own analysis on this pooled set of data. That's basically what a meta-analysis is. So here brought some history, a description, and the goals of meta-analysis. Again, I'd like to start with a citation. Meta-analytical thinking moves researchers beyond their own study and views their study in the light of the greater body of literature. So I will come back to this citation when I finished the slide. First of all, I want to give you a bit history of meta-analysis. It was introduced by Jean Glass in the early or mid-70s. Meta-analysis basically describes the use of quantitative methods to summarize empirical prior studies. So as I already said, those are primary studies that tested similar hypotheses, or at least provide a similar set of data. So now let's move to the right-hand side of the slide, the description and goals of meta-analysis. As more primary empirical research may not necessarily decrease the uncertainty of a particular topic, for example, as findings might be inconsistent or models may not be comparable, a comprehensive analysis of empirical evidence available may be necessary. So let's assume We want to investigate whether an effect exists or whether it does not exist. And we look into literature, right? And prior research found disparate results. So they are arguing whether it's true that this effect exists or whether it does not exist. So then meta-analysis could be helpful because you gather the data from the primary studies. You look at the results, so you see, well, they are disparate, and you try to find why they are disparate and try to find an overall conclusion. So that is the purpose of meta-analysis. It's to integrate findings and determine how large an effect on averages, so on average, considering all your primary studies. It's about determining factors that are related to the effect size or specific study characteristics, which might explain why the results are different. So why those researchers who already conducted the studies found different things or different results. And then you can draw your overall conclusions. So that's exactly what the citation at the beginning of the slide meant when it said it views your study results in the light of the greater body of literature. So you expand your horizon, for example, because those studies only interpreted their results in the light of their own study, so in their context. And as you're pooling these data, you have the ability to draw conclusions regarding the overall sample size of your primary studies. So this statistical combination of effect sizes or results, basically the results we combine are the effect sizes in primary studies, is called the pooling of data. The unit of analysis are, well, the results or the resulting effect sizes in these primary studies. And these primary studies are or should be independent, and you want to form a new comprehensive database. So you pool your data, and this pooled data is your new database on which you perform your meta-analysis. So you perform a meta-analysis, which is basically a statistical analysis of the new comprehensive data set you gathered, and you want to integrate the findings and derive overall conclusions on the basis of your pooled data set. So there are two rough understandings of meta-analysis, and we focus on the right one. So one understanding of meta-analysis is an individual-based meta-analysis, which is basically rooted in the field of medicine or psychology. So this understanding of meta-analysis is called, for example, individual patient data. So a meta-analysis based on individual patient data. We talk about patient data because this form or understanding of meta-analysis is rooted in psychology. And the idea of meta-analyzing individual patient data is to draw or conduct a synthesis based on this primary data. So you really collect the raw data, not the results of the primary analysis, but you collect the primary data from individual primary studies, for example. We are talking about an aggregation-based meta-analysis. So that's the understanding of meta-analysis we focus on. It's on aggregated patient data or aggregated data, basically because we don't look at patients, but we look at primary studies. So we synthesize studies or study results instead of real data in the primary studies. So what we are interested in are statistical outputs or statistical measures, which are the subject of our analysis. So we extract numerical estimates from the primary studies and combine them to our meta-analyzing data set. So this is a deep dive on the subject of analysis in meta-analysis. So what do we analyze? What do we really collect out of these primary studies? The unit of analysis is an effect size, for example, such as a summary statistic reported in primary studies. The definition of this unit of analysis is it is a standardized index for direction and strength of a relationship between two variables. So this effect size we collect tells us, for example, how the relationship between two variables of interest is. There are three families of potential effect sizes which we might collect from those primary studies. First, we might collect mean differences and standard deviation effect sizes such as Cohen's d. Another family of effect sizes refers to binary data such as the odds ratio. The third family of effect size is based on correlations such as the Pearson correlation coefficient. By the way, the Pearson correlation coefficient is the correlation coefficient which is most popular in meta-analysis. The requirements on effect sizes are as follows. They should indicate a direction. Thus, correlation coefficients are very favorable, so we won't use, for example, an R-square from regression analysis in primary studies because they don't indicate a direction. Effect sizes should be relative independent from sample size, and they should be standardized in order to compare them across studies. Further, it would be favorable if effect sizes could be transferred to some other effect sizes. For example, imagine that some primary studies report standard deviations and others report variances. So we could collect these effect sizes and transform them to have the same base of effect sizes. Here are some pros and cons of meta-analysis compared to primary analysis. On the left-hand side of the slide, we see the pros, or the advantages, which are really kept short. The cons are kept larger, as I also provide solutions to solve these problems or to dissolve these disadvantages. Let's talk about the advantages. It's good in meta-analysis that more data can be analyzed, and we have the possibility to think outside the box, to broaden our horizon as we look at very different studies, or as we look at a lot of studies which might have been conducted in very different contexts. And as we gather or pool these studies, we have the possibility or the ability to look beyond those single contexts and to draw, as I already said, an overall conclusion. Now let's talk about the disadvantages of meta-analysis. There is the threat or the risk of mixing very dissimilar studies, so it's called mixing apples and oranges. The problem is that synthesizing effect sizes that are not meaningful might be not good for our study because we aggregate those effect sizes, right? So one possible solution might be to form subsets of our studies according to moderators. So to create homogeneous studies and to analyze them subset by subset. There's also the risk of publication bias. So as we're doing a meta-analysis, we really require a lot of primary studies. And there's the risk that some studies might not have been published yet or will not be published. For example, studies which found no effect and the authors thought, well, that's not a publishable study. But in meta-analysis, we really should have all studies, and we need a lot of studies. It's better to have more than less, right? And there's the problem that you might not integrate all studies available out there in your meta-analysis. Well, it's difficult to solve this situation, But you could reach out to the authors of a certain community. For example, when you're doing a study on collective intelligence, you have the possibility to look into prior literature, look into the field, write authors who presented their work or working papers at conferences, or those authors who have already published something in this field, telling them that you are doing a meta-analysis and asking them for unpublished work. Finally, as you gather a lot of studies, you might run the risk of including poor quality studies. It's called garbage in, garbage out problem. And you should only include valid studies. So for example, when you're collecting or gathering studies that performed or conducted an experiment, you might only include those studies that have a decent randomization in their experiments to make sure that your meta-analysis has a fairly good quality.